It's okay. It's
Okay,
clear.
okay, thank you.
And thanks a lot, sir, for joining.
No problem. It's a privilege. Yeah. So I have always interaction, right? You know.
Yes, yes. Okay, we are live. I think we're live now. And thanks a lot, sir, for joining.
No problem. It's a privilege. Yeah. So I have always.
Okay, sir. Then we should start. It's 2 p.m. Welcome, everyone. Today is our 25th webinar. I'm very happy. I'm not thought about that one day we go as on 25th. Now we decided to keep moving and keep going this platform and this type of session. We're very fortunate to have Dr. Manish Pandey from ICRISAT. I know him very well, his group, and I admired a lot about their publication and their work on genomics, and specifically when someone work in some species and variety which very few people are working and which has very biofortification value, nutritional value. So I'm fortunate Dr. Manish working on groundnut, peanut, chickpea, pigeon pea, this type of crops. I request Soma to introduce Bioingene little bit and then our speaker. And for today, Shoma will be the host and coordinate the webinar full session. After the webinar, we collect all questions and ask few of them to our speaker, and we give the password for the certificate collection. And I hope all registered participants will get the link for the feedback and certificate collection. Thanks for joining with us. Thanks, Dr. Manish sir, for giving us an opportunity and joining our webinar session. Shoma, please introduce our Bioingene and sir.
Hello, everyone. Welcome to Bioingene's webinar number 25. The webinar series has been designed to build a platform from where plant scientists can present their research to the whole world, and future scientists can gain perspective and inspiration. The appreciations that we receive from our global viewers after every webinar keep us motivated. We have started a new series called the Bioengine Live Discussions. This is mostly a peer-to-peer discussion section dedicated to research in the field of life sciences. This new series is also a part of the Bioengine initiative. The discussions include important aspects of our research life, like research planning, conceptualizing, methodology designing, manuscript writing, finding appropriate journals for publication, scientific communication, national and international fellowship for PhD and postdoc research.
Yes, sir.
You are welcome to join this initiative and participate in these discussions on our
Yes,
YouTube
sir.
channel, and also post your ideas on the topics you want to be discussed in the live sessions. Please visit our website to know more and register. Now, coming back.
Okay, sir. Sir, open. Yes, sir.
Mute. Keep your mic on mute. Hello, Pallavi.
Okay, sir.
Okay, thank you. All right. So where was I? Now coming back to today's event, today is our 24th webinar of the series, as I told you, and we welcome all the viewers who have joined us from around the world. After attending today's talk, you can apply for a certificate of participation via the feedback link and password. We have already emailed the feedback certificate application link to the registered participants. The password will be provided multiple times in the YouTube chat box during the question and answer session and the interview session. Please submit the feedback form, which is mandatory to receive a certificate. When you fill out this feedback form, please mention the full name of your institute and the full institute address. You can collect your participation certificates after 2 days from our website. Also, Bioingene does not send certificates through email. All right, so please visit our website to register for upcoming webinars. You can connect with Bioingene via Facebook, YouTube, Twitter, and LinkedIn. Now, let me introduce our speaker for today's webinar. Today's webinar is on Translational Genomics for Achieving Higher Genetic Gains in Groundnut by Dr. Manish K Pandey from ICRISAT, India. Dr. Manish K Pandey completed his PhD in plant genetics from Osmania University while working in the ICAR-Indian Institute of Rice Research, or known as IIRR, Hyderabad, India. He gained postdoctoral research experience at the University of Georgia, USA, for a period of 2 years. He is currently leading the Groundnut Genomics group at International Crops Research Institute for the Semi-Arid Tropics, or all of you know as ICRISAT, Hyderabad, India. He is also an Adjunct Associate Professor at the University of Southern Queensland, Australia. His key contributions include reference genome sequence for diploid progenitor and both the subspecies of cultivated tetraploid groundnut, gene expression atlas, low to high density SNP genotyping assay, diagnostic markers for key traits, quality control panel, marker assisted pyramiding, and genomic selection in groundnut. Through genomic based breeding, his team developed two high oleic groundnut varieties which are released for cultivation in 6 states of India. He has published more than 150 scientific articles in various journals of international repute, including Nature Genetic, PNAS USA, and Molecular Plant. His efforts have been recognized at the international level, and he has been invited to deliver presentations in several international conferences to review the proposal for international funding agencies. He is also collaborating with large number of scientists at international level. Recognizing his contributions, he has now been inducted in many scientific academies of India, namely the Associate Fellow of National Academy of Agricultural Science, or as you know as NAS, New Delhi, the Telangana Academy of Agricultural Sciences, the Andhra Pradesh Academy of Agricultural Sciences, Andhra Pradesh, and a member of the National Academy of Sciences, India. He was also among the top 2% of highly cited researchers in 2019, compiled by the Stanford University, USA. We are highly honored to have him as our guest speaker for today's webinar. It is my request to all our viewers to enjoy this opportunity to listen to him speak about his research. I now request Dr. Pandey to please share his screen and begin his talk.
Thank you so much, and thanks to Dr. Majumdar and really Bioingene Group to invite me.
The sound is breaking.
Little bit, yeah.
Yes.
Sometimes it's up, sometimes it's down. Yeah.
Is that fine now?
Yes. Now fine.
Okay. Okay. So basically, it's a thanks to Bioingene and Dr. Majumdar, and of course, nice introduction.
No, sir. Your sound is gone. You can remove your headphone. Yes.
Without microphone?
Yes. Yes. You go for
Try
without
that.
your phone.
Yeah.
Okay.
It's okay. Fine.
Maybe microphone problem.
Yes. Yes. Okay.
Okay. So let me share my slides. Okay. So basically today's seminar, as Soma mentioned, that it's on translation genomics for achieving higher genetic gains in groundnut. So basically I will be working, I'm working basically since 10 years now in groundnut. You can say basically after PhD, I have spent all my 10 years working in groundnut. So I will take you today to a journey from where we started in 10 years back, where we stand now, and what we think that we should do to make more genetic gains in groundnut.
March, April, May, June, July, August, September, October, November.
Shiva, please stop counting the months.
Okay, okay,
think
sorry.
slide is struck.
Little audio problem.
Yes, yes. Sir, we can't,
Oh,
s-
it's okay now. I think slide was struck.
okay.
Okay, so yeah, so as Soma mentioned that I work in ICRISAT, and you can basically you can see that ICRISAT is part of CGIR, and CGIR has many sister institutes. ICRISAT is one of them, and that basically in India only one CGIR center headquarters, and that is ICRISAT, that is based in Hyderabad. So, and really it is a good platform, ICRISAT, because we basically serve as a bridge between the advanced research institutes and the national partners in different countries. So if you say that we do very upstream science, no. If you say that basically we do very downstream applications with farmers, no. What we do basically that most of the scientific information we just work with advanced institute and NAS partner to convert them into a technology or in terms of a product, and then making sure that it reaches to the farmers. So basically a bridge between these two kind of organization, and we do conduct research in these areas where we can fill the gap. Whatever we work at ICRISAT, I would like to mention that it is all international public good. That means whatever information, products, technology we generate, it is freely accessible to all, everyone across the globe. There is no limitation or any hindrance in accessing those technologies. That is the one good thing that really gives encouragement that you can collaborate with anyone. Basically you are taking information or basically working with gaining knowledge from everyone, and also you have freedom to disseminate your information technology to everyone. That's a really beautiful platform, very encouraging. At ICRISAT we have good platforms, whether it's phenotyping fields, experimental fields, and also lab sequencing, and we have like we have even like say WGRS and other basically high throughput sequencing. It's not good to have basically very big sequencing labs. Of course, we need to have only those relevant instruments in our labs, which from where you can generate cost-effective sequencing data or genotyping data. Otherwise, it will not be irrelevant in that one. So, and we have server as well. Of course, you all are aware that when a lot of data is generated, then of course we need to be more cautious. So, in that case, let's go to next. So, ICRISAT works in 6 crops. Why I'm mentioning here that I will be giving examples from groundnut only because I work in groundnut. But in general, the principles and the approach, it is same in all the crops, that methods and technologies we are using. So you can relate to whatever crops you are working. Of course, there is customization and something maneuvering you have to do in different crops because the resources, they are different in each crop. And so these are the 6 crops: pearl millet, sorghum, finger millet, chickpea, and groundnut. Groundnut basically in different parts of the world we know and consume in a different way. Like in Asia and Africa, if we say, it is more of not only just like a confectionery item, and that like in Western world, like in US and Europe, but it's actually a very good source of nutrition in India and Africa as well. And you can see many of the products. So of course, any crop in market, if it has to survive, definitely market has to play a very good role, because industry is the one that provides different kind of items, different kind of like preparations to consume to the consumer. So basically it's a consumer driven agriculture, and we have to see that whichever crop we work, maximum beneficiaries should be the consumers, then only that crop is going to be relevant. And really, if you have seen in last 70, 80 years, you might have seen how the crop relevance has changed from green revolution earlier, like finger millet and millets, to then rice and wheat agriculture, and now we are again coming back to the small millets and others in our daily food habit. And in India and Africa, this was very routine basically in our. I remember that even my childhood, basically we were more relying on the sorghum, finger millet, and pearl millet. But rice wheat has changed. Maybe we are going to back basically finger millet and something, because those are more nutritious. So now what is happening actually? From quantity, we are going to the quality. From eating not a high volume food, if you might have seen from yourself, the consumption is becoming less, but we are trying to eat more nutritious, and that should be the approach. So the crops which are more nutritious are going to be surviving in near future and with less water. So we will be discussing, yes, of course, groundnut, it has different applications, right, from cooking oil in Africa, India. And one more important thing I would like to mention here, even in India, if you might have seen, like basically 10 years back, more 70% of the groundnut that were extracted for oil, for cooking oil. But what we have seen, that industry has played so much good role that they have provided different items, food, they have diversified the preparations, that now as on today, around 50% of the product produced that goes for oil extraction, and rest 50% goes for industry. That's a really a different sign altogether. So basically groundnut is in transition stage, and there is more demand for protein and food. And you can see in Africa, like plumpy nut and other things, there are many applications. It's a really good source. Okay, now the basic question comes, you might have seen, like what is going to do, climate change is going to change, every country is going to be suffering losses? I don't think so. The countries which have more cold, of course they will be gaining some benefit out of it from this climate change, rising temperature especially, where they can have possibility for more new crops. What other, like especially the red ones, if you see Africa and Asia, we will be facing problem for most of the crops, that because of rising temperature, and if it is clubbed with drought and water problem, then definitely yield reduction is going to happen. The worst scenario would be if called quality and nutritional quality also compromised, then this becomes really serious for Asia and Africa. Okay, this is a paper that where we just summarize like how these four pillars that make really realize high genetic gains. Those are the maximizing genetic diversity. That means the pop lines in breeding we use, crop improvement, those should be diverse, so that we can have high genetic diversity in our breeding material. Second is improved genetic enhancement. You might have seen that that new genetic improvement approaches are coming up. Most recent, you might have been hearing about genomic selection, genome editing, such kind of approaches. So definitely they are coming up new, and we are scientists are integrating them into programs. Third one is improved operational efficiency. That means basically not only that scientific things, what operations we do, whether it's analysis, whether it's a field data management, whether it's a field design. So sometimes maybe we don't give those so much importance to these, but actually these are very important component. We should actually now, because we are generating now large scale data, and when high throughput large scale data comes, somewhere you have to manage the data and ensure that those data sets are connected with each other, so that whenever you want some information, you can quickly access those information for decision making. So that's very important. If we had earlier like less information, you can put in Excel sheets and all those things, you can manage, you can take decision. But now the situation in condition is changing. We are having large data sets. Okay, now efficient seed system. I think the more we talk, it's less in this one. So really, it's from country to country, challenges are there, because providing quality seed to the farmers in time, it's really challenging. And in many of the countries, private sectors are playing a very key role, but not in all the crops. Private seed companies, they are in the crops, basically mostly hybrid crops, where they can play a key role. But like groundnut, definitely the government agencies and public private, those agencies have to play crucial roles. Once you put all these things then only that whatever you improve the seed genetics, basically that will be realized in farmers field. So basically we have to take care of all these components. You just can relate to ourselves as a human being. We have a set of genes, we have our set of genes, but you can see that even brothers, sisters, and even the nearest one closest, they have different kind of phenotypes, different kinds of performance and other things, because there is a lot of things that happen throughout our growing up, that we see changes and efficiency. So that is how we have to be, basically with our human life. Yes, food habits, so that's very important. And here genetic gains means that you have to ensure that whatever genetics you are improving in the lab, that should be realized in farmers field, otherwise there is no use of that. I will be discussing today how we are using actually different modern approaches, how we have shifted from like previous to the modern approaches or methods, and how it is helping us in improving the genetic gain as well as in faster manner. So we will go here. This is a recent paper, although this is we have published in Groundnut, but it is relevant to all the crops because we have tried to make it generic. So basically, you can see genetic resources. Of course, you need to do anything. You need some resources, genetic resources especially. So gene banks, yes, they have, but how to use it? That's a challenge. Breeders are very much comfortable in using the elite lines because they don't want to take risk of genetic linkage drag. So how basically we can take limited risk, how to maneuver basically our genetic improvement program. So we need these genetic resources, different kind of populations, and then of course genomic resources. These are the must. Once you want to implement modern breeding approaches, then definitely we need these resources. And we will go one by one to these resources. I will not go here in detail, but because slides are on this. And then applications. So whenever we say actually in genomics, most of the times you discuss about marker state breeding or any molecular breeding product or linked marker like that. But actually genomics is not all about just developing associated markers for a particular trait. It can be used for different things, like you can assess basically the duplication in gene bank. See, we collect germplasm from farmers and land races and other things. We don't know actually if it is the same, whether our fellow scientist has collected and I have collected. So really you can check it on, you can have duplication. Then candidate gene discovery marker. Yes, this is the most talked about this one. And then you can have marker state breeding, genomic selection, and like wide hybridization where you can eliminate the linkage drag. Of course, in some crops, hybridity conformation like rice, wheat, maize, where you can see so much variation between parents and you can identify F1 plants very easily in the field. But in some crops like even groundnut, it's not very easy. As many of you may know that for groundnut, hybridization happens aerial. That means above the ground. And then peg goes inside the soil and forms the pod. That means, and the variation you can't see on the area, basically in the shoot, much variation in groundnut. So most of the variation comes in pods and seeds. That means even for identifying true F1s, for breeder, it's challenging. And in even the best breeding programs, you can find possibility of 10 to 20% of selfing in that case. What I mean to say that if you don't basically confirm the F1, then your genetic and breeding population are going to be biased. So, and of course, that will lead to your impacting the genetic studies as well. Seed purity assessment, yes, we will go in that seed system. Of course, this is a very good area. I think very less export. If you ask anyone that which variety is occupying how much area in a country, that information is coming from one or two sources, but not actually from the field. But if you can use markers to check that actually what kind of varieties farmers are growing, you can check real-time samples. Definitely you will get accurate data that which variety is occupying how much area. So that would be the approach. Today, let's go through these 10 important aspects here, and then all the things are interconnected, and then I will be putting one and one. Most of the examples are from groundnut. In one or two places where we don't have much development in groundnut, I have put from different crops. That means we are working on that, and in 1 or 2 years or 3 years, so we will have examples in that area. First is genomic and genetic resources. As I mentioned earlier, what you can see basically that sequencing, right? I think everybody now know that sequencing cost is no more an issue, right? We have come a long way, and really last 1 decade we have seen that how different technologies they have emerged up, different machine they have come up with different high throughput data, reads, length, and all those things. So basically what I mean to say here that now sequencing data generation is not an issue, very cost effective. You have to work with appropriate service provider. Definitely you are going to get very much affordable sequencing data. Because of that, what you can see is that if you say 10 years back, nobody was taking risk for developing reference genome, high quality reference genome for complex crops, like say wheat, maize, basically like even groundnut, like tetraploid. So where you have different ploidy levels, right? It was challenging because we had limitations in terms of sequencing data, length generation, and also the tools which can combine these data sets into a good quality reference genome. And you can see like basically PacBio, Dovetail, and 10x Genomics and BioNanoMap. So basically coming up different technologies, and now you can develop really high quality genome. If you have money, you just talk to company, they will arrange everything, including analysis and all those things. So some basic analysis, and of course downstream you can see. So what I mean to say here, if you want to develop reference genome, you need not to be worried about. There are already companies, they have got that kind of expertise, that if you tell that what kind of genetic material you have, what kind of quality you want, what kind of genetic resources you have to check the quality of the assembly, and then definitely you are going to get high quality genomes. That is why you see that most of the genomes coming now, they include not only that basically PacBio, but also high C technology they have been integrating to make sure that the genome quality is of high quality. What we have done basically with national partners and most importantly advanced research institutes, developing because groundnut is a tetraploid genome, it has two genomes, A and B subgenomes. So we have first sequenced the diploid progenitors. That means Arachis durarensis and Arachis ipiensis. And then after that, we have sequenced tetraploid genome. What you can see basically here, if you see in the table that you have Tifraner, that is Hypogea subspecies, and then you have Shitoki, that is Fastigiata subspecies. Why I'm mentioning here another genotype, that is Fuhuasheng, basically that is also Fastigiata. And you can see that High C and PacBio had been the technology that is being used. One important thing I would like to mention here, these two genomes are of different subspecies. When I say subspecies, in like mostly like 70 to 80% area, that is Spanish bunch in India that we grow. That groundnut plant, if you see little bit straighter, that is Spanish bunch. And the another one is runner. You might have seen in US and Europe, basically the plant that creeps basically, and it takes basically big area. So that is runner type genotypes, and that has less area, but again it's long duration and high yielding. The Spanish bunch is straighter, and then it's short duration, mid duration, and then little bit low production, low yielding than the runner. So basically what here we mean to say that we have a reference genome for both kind of subspecies which are grown today in world. Okay, now the question is, have we achieved that we can stop sequencing genomes and say now we don't need it? I will say, if you see the groundnut how it is, so you can see in the tables that we have genus Arachis, and then you have different sections. In that one you have Arachis sections. Then within Arachis section you have 31 species, and in those 31 you have 6 kind of genomes. Why the yellow highlighted what you have seen, A, B, and AB? means those are sequenced, and the DFK, basically those are the genomes not been yet sequenced. So, and this is, I'm talking only Arachis section. I have not mentioned the other sections you see top. Those are also needs to be sequenced. What you have also been seen that in table in ploidy, if you see, there is a big ploidy difference also, diploid and tetraploid. What that means, in breeding it's not easy, that's very challenging to bring genomic regions or making crosses when there is a ploidy difference. So there are different routes through you can make some like synthetics and others, and you can make crosses, then only you can bring in diversity from other species. What I mean to say here that at least the cultivated ones, the one we are growing, we have genomes. So basically we can do any kind of high quality or precise analysis using those genomes, and maybe we can continue in long run. That you can see second right part where we are talking about genomes and pangenomes and super pangenomes. That means you all one genotype, one reference genome cannot represent the basically genome for all the germplasm that exist here. Basically, for each species, if we have a one basically high quality genome, that would be good. That is why pangenome and core pangenome, I think super pangenome, these are going to be critical. We have just completed these basic genomes here, and definitely we are trying to work with international community to move to this direction to the core genomes and pangenomes. What will happen basically in the long run, maybe I see from 10 years or so, maybe we will have sequencing data for all the germplasm of groundnut that exists. Challenge would be how to analyze and how to make good use of this data in our breeding program. So that would be challenging. Just I would like to mention here one paper that came in Nature, that is on 25th November, and it is coming from wheat. And what you can see that they have compared, they have justified basically, if you see why you need multiple genomes. For example, like how, like in human genome, just I will give an example. If you have one human genome that is from Europe or any place or Africa, how it can represent for different continents. So basically, you need to see what kind of genomic diversity you have within the population, and you need to identify the best model, basically the ideal genotype that can present maximum I say reference genome. That is really a very good question, and that is why this paper tried to answer, because they have here compared 10 chromosome means pseudomolecule level high quality genomes, and also 5 scaffold level assemblies. What you can see basically in total, they have compared 15 genomes and try to see why different genomes are required, because each genome they were seeing that either they have some large integrations, some translocations, inversions. That means if you have one reference genome and you are comparing your genotype, it may not match up. Some of the sequence may be deleted, some may be added there, but 100% like basically when you align your sequences, it is not going to be perfect analysis. So something will be missing. So that is what this paper is about. This is really very good paper, and I think those who are interested should read it. It is really very good paper. Okay, now coming from genomes to another one, that is this gene expression atlas. So we try to do here basically landscape profiling for those expressed genes for different tissues. We collected some. But the right side you can see it's for Fastigiata. That is the Spanish bunch, the variety we grow here in Asia and Africa that comes from. So we developed this one here at ICRISAT. The left one you see is from Hypogaea, that is from Tifferer. Basically, that is from Runner Group, and that is done by our colleagues, our collaborator from University of Georgia. So what you can see, not only genomes, this gene expression atlas, they are also developed for both these subspecies. That's a very good thing. So suppose if you are doing some analysis and you got some genes, you want to check whether it's a tissue specific or not. These databases are available. You can check, okay, whether it's a seed specific, whether it's a tissue leaf specific or not. So they like those kind of information, it may helpful. Okay, so because the mandate here, what we have ICRISAT. So this is the work, what you can see that we have been doing with our advanced research institutes. As I mentioned earlier, that we work with advanced research institutes, and also we work with our national partners. So basically, these resources are very much required for deployment. So we worked with advanced research institutes and NAS partners to develop those resources. Now, what we worked basically to develop those intermediate resources, different cost-effective genotyping assays, so that the breeding and genetics community can use in groundnut. What kind of assays? So basically, in 10 years, we have started. Actually, I have not mentioned that one. That is page. When I came here, we were genotyping on page, and really it was very like tedious, laborious, and then really very tedious to do. That you cannot run more than 3-4 gels in a day, and really it was very tiring. We have shifted now from SSRs and DART we use when we did not have genomes, but we are not using right now. CASP assays, yes, whenever we develop some marker assays, CASP is the one that we have been highly utilizing. GBS, I will say again, basically when we did not have this 58K SNPs, the one in yellow you are seeing, the array, we were using GBS, otherwise GBS has some serious problems and missing and other things. So basically, I'm not big fan of GBS in that way. 50K SNPs, those are the good ones. They are working well for mapping purposes and other things. So WGRS, yes. So it's again, if you have money, you are rich, you have a lot of money in your pocket, you can go for WGRS and then you can generate data in mapping. Then 10K, 10 SNPs panel, I will be showing that one. So basically, here we are saying we have two kinds of uses. One, we need large number of markers, and then we will be genotyping on say 100, 200, 300, 400 breeding material or genetic populations for mapping. The other use is like that we have a small set of markers, say 10, 20, and then we will be screening them in thousands of lines. Those will be trait linked markers or QC or something like that. That will be used large set. So one, we have less number of SNPs, large number of samples. One, we have large number of SNPs and a small number of samples. So that is how we need to customize these assays, and that is why we are now developing one intermediate assay, that is mid density assay with 5,000, and now we are trying to clubbing the application for both the things, like basically I will be showing in those slides. So this is the like basically you can say journey from SSR to SNPs. Now we are 95% using SNP data in our breeding program currently, and it's very cost effective how we have made it. I will take you to those slides. So if you see like high density genotyping, as I mentioned, 58K SNP chips that we developed, it's really very useful. There are 58,000 SNPs on that. And if you genotype for genetic diversity, trait mapping, genomic selection, marker-based varietal adoption, these are good. This is working good. We have tested it. GBS, again, if you don't have any option, I will say you can go for GBS. I'm not a big fan of it. Or you can increase the basically sequencing depth so that you can have more data. And whole genome sequencing, yes, in some cases we are using whole genome sequencing. I will tell you for large population, large number of samples, it's not so easy, it's very costly. So like that way. Okay, coming mid density that I was talking about, that's really very important. This is new, I think in only selected crops, maybe 1 or 2 crops it is available. So basically in next 3-4 years, I think many crops may be having such assays. What we are doing basically, we are keeping 5,000 SNPs and also including here 160 trait associated SNPs. What that mean? That we will use it for genomic selection purpose. So suppose if you are doing genomic selection, what you can do, you can select some 10-20 line based on genomic selection, and if you have trait linked marker, then you can imply those selection criteria, and then you can select those trait as well in that one. And it's very cheap, basically less than $10 per sample, and definitely then you can use it for various purposes. The high density, I said, it's mostly available about $60 per sample or so. So those are the costly one. This is just $10, less than $10. It is going to be more cheaper, because when competition comes between different service providers, definitely the consumers are the beneficiaries. So okay, here we are seeing that option. Okay, if you want to use foreground selection and background selection in your marker background breeding program, of course you can use it. So this is not that much costly. Genetic purity, actually this is a big concern. It is a big concern in many of the breeding programs, although because those seeds are handled by technicians in field, laborers, and then packaging. So basically that seed goes from hands to hand during the processing in a single breeding program. So that means error can happen at any point of time. The breeder needs to maintain genetic purity in their breeding program, the parents they are using. Otherwise, there will be problem basically in the further generation. So, and marker based varietal adoption, yes, we are looking for it, that we want to assess the actual varietal adoption, not that based on some record somebody has recorded. Basically, it's based on the marker real time check that how much this variety is being adopted in a particular region. This is low density. So basically like F1, as I mentioned that in breeding program, F1s needs to be checked in many of the crops. If you can see it, I will just leave it on the breeders. If they are convinced, they are 100% sure, they can go for it. But what we are encouraging that QC should be used. F1 definitely should be confirmed. Otherwise, your breeding and genetic population will be biased. So what we have done basically, we have developed a QC panel that is with only 20 SNPs, and we have included here from ICRISAT Asia, ICRISAT WCA, ICRISAT ESA, and also in India ICR-DGR. That means we have genotyped all the founder parents, means the parents these institutes are using in the breeding program, and these 20 SNPs can differentiate any of these cross combinations. So in these 20 SNPs, so what we are doing at ICRISAT, how to use it? As I mentioned that for 10 SNPs at Intertech, you can get data in 100 rupees. That means $1.5 you can get for 10 SNP data for a single sample. And that is to without DNA isolation. That's really cheap. So if you define that, okay, you are going to use next 2 years, these 20 parents, 50 parents, then from these 20, you can pick those polymorphic SNPs, and then you make a set of 10 SNPs. Even the breeder is even more like basically taking interest, even like we are doing at ICRISAT. Even here, right, for this I'm telling about this post-rainy season, our breeder is making crosses using some 15 parents. So basically 7 SNPs only, out of that 7, at least minimum 4 SNPs are differentiating those combinations. So 3 SNPs are remaining. In that one, you can have trait associated SNPs in that. What I mean to say that you can use it in different ways. 10 SNPs, cost remains same. For 10 SNPs per sample, you are going to pay only 100 rupees without DNA isolation. Why this without DNA isolation? Because we are working in ICRISAT, and as I mentioned, we have our breeding locations in Africa, and even in India. If you see the breeding centers in remote areas, if you see, they don't have a DNA isolation facility. And so that means if you remove this hurdle of DNA isolation, you just chip the sample, like punch it, and then send directly from field to the Intertech company, and they will be genotyping it. So it's very simple. No, you don't need any lab. That's what I mean to say. Earlier I was saying that for sequencing everyone should not have lab. Now I will say even for DNA isolation everyone should not have lab, and you can use this facility for genotyping. So that is really promising, and it is helping really very well for our African breeding centers, and also we have seen adoption in India as well. Now coming to the gene discovery and diagnostic markers. Of course, if you want to use marker selection, marker backcross breeding, definitely you need linked markers. And also, even if you want to deploy genome editing kind of approaches, also you need to have access to the genes whose function is known that you are going to edit for crop improvement. So basically you need gene discovery, and really sequencing has helped a lot and the analysis. If you see, I have just listed some papers also here. Interested people can see, they can read it. So you have now different options of mapping. Now you need to see what resources you have, what kind of genetic populations you have, and then how much money you have, how much you can afford, and then basically you can choose the genotyping platform, sequencing options, and then you can go for trait mapping, right? I will be presenting here how we are using ICRISAT, especially in groundnut, that we are getting done in very less time and then more precisely. We have here different kind of genetic populations. Earlier you might have seen biparental, that was normal. Now we have actually 3 magic populations. That is really very diverse material. One for aflatoxin, one for drought tolerance, and one for disease and nutrition. Similarly, we have nested association mapping populations. So we have here like Spanish and Valencia, 2 different kind of groups. So basically we have made separately these norm populations for mapping. Magic and norm population actually not just they serve for mapping, they are diverse breeding material. So really for breeding also it's a good, you can select very good material out of it. Association mapping, it's everyone knowing is it's a normal. Basically, although concept is coming from animal sciences where you can't have such kind of biparental and other populations, so association mapping is only the option. And in plants also it is serving very good method, especially in those plant species those are perennial or something like that. So really that's very useful, this approach. Okay, if you see groundnut which traits we are interested, so you can see some of the traits that we are targeting. That is basically we have based on Africa and India locations, Asia, these locations we have started working. So these I am not listing here this breeding that we are doing breeding for this. I am just talking about trait mapping and then making available diagnostic markers for these traits. Okay, now let's come to the sequencing based approaches. So one approach is you can see trait mapping through sequencing of complete population. Suppose if you have recombinant inbuilt line, you can generate genotyping by sequencing data, or if you have money, you can go for whole genome resequencing. That means WGRS data, you can go 5x, or it's up to you, money, right? But even like how much it is required, you can see based on the genome size, you can estimate it. The second approach, you might have heard of bulk segregant analysis from here in I think 90s, and then where you pool samples basically like resistant and susceptible pools. So QTL-seq means suppose if you have real population for disease resistance, you can pool DNA from 20 highly resistant and from one that is one pool, and another pool susceptible you can make by pooling highly susceptible 20 genotypes. That means you have a sample susceptible bulk, you have resistant bulk, and then also you can sequence 2 parents. So you have 4 samples for QTLC. If it is not a normal genotype, it is one parent is mutant, then you can say mute map. So it's just basically sorry, it's a basically a terminology SecBSA. If you don't get basically much from peak, basically you can't get, then you can go genome wide, the BSR-seq, also like from based on RNA. Indel-seq, not only SNPs if you go for indels. So basically it's a terminology. What I mean to say, once you have such kind of population, now these are the approaches you can complete. If you have everything ready, phenotyping data on the population samples, you can sequence and perform analysis. Within 6 months, you can get results on genes and markers. So that is the like advantage. We have used some of these approaches very heavily. I will show one by one. Here we have used with our national partner DGR, that is for eastern blot resistance, identifying genomic region and candidate genes. I will not go in detail basically, but I will be showing those examples. And here we have used genotyping by sequencing. You can see this paper here. We have gone for disease resistance and we have used whole genome resequencing, basically WGRS. Yes, of course, you can have basically very good mapping, very good. You can go to the genomic regions, you can get genes, then you can develop caspases. Really, it's very helpful. But of course, it is costly. Now, this is the example. Now, shifting to pooled based sequencing that I was mentioning, that is QTL-seq. You can see here that it is for rust and late leaf spot. So really, we could develop markers from these, whether it's a lab, really specific simple markers that you can run in lab, or CASP assay that I will show later that we are using at the Intertech platform. I was mentioning $1.5 cost, so we are using that one. So this is like basically here rust and late Lepis. But we had data, we were struggling to find genes and really reliable markers. We completed this task in 6 months after sequencing. Here is another example that we have done with Chinese colleagues. And here the trait you can see is selling percentage. And the another side you can see bacterial wilt, that is a disease. You can see how disastrous that disease is, and that is in China. Here we used basically a modified approach, both the parents in the analysis one by one. And original QTL-seq approach, if you see, that is based on only using single parent. So here we used basically two parents one by one to see that what kind of results we are getting and differences. So we end up developing markers for both of these traits. We have recently finished for fresh seed dormancy. So in groundnut, actually this is a serious problem, especially in Spanish, the one we grow in India and Africa. Suppose during harvesting, if it is delayed for 1 week and it is rain, I think 70–80% of your pods or seeds you see germinated inside the soil. Really a huge loss. And really it is very, and whatever remaining pods will be there, their quality will not be good. That means you are losing a lot. So we could develop marker for this one. So QTL-seq approach really very useful. You can see third one. Currently we are doing multiple for multiple traits, and in future they will be coming up. I will not go in detail, but I will touch here one of the most complicated trait, that is aflatoxin contamination. You might have heard that apart from maize, groundnut is one of the crop that is having, there is a standard set in US and Europe for export from India. And it is good, not good for health. I will not go detail in all those things. What we are trying to do it, that we are trying to understand these three kinds of resistance mechanism. In vitro seed colonization means if the pathogen comes in contact to the seed in post-harvest, then it resists from the infection. Second is pre-harvest contamination, that is field resistance. That means in the field, if pathogen comes to the contact of pods and seeds, then it resists. The third one is aflatoxin production. There are also some genotypes, even the infection happens, they resist producing toxin. So that means what we are doing, these resistance mechanisms are present in different genotypes. What we are trying to do with different approaches, these jivas and then magic, by crossing these things, we are trying to bring all these three resistance mechanisms into one. That means first, it should not allow to get infected in field. If it gets some infection, it should resist during post-harvest. And also if it increases, then it should resist toxin production. So that is our approach. Let's see how much we will be successful in coming years. So this is just in short. I will not go in detail because this is a very big story in that. I would like also to touch about peanut allergy. I think US and Europe, you might have heard, even in Australia, that's a huge population. And when last 2–3 years, we wanted to see that what kind of, what level of actually allergens, proteins we have in our groundnut accessions, varieties that we eat. Although our Asian and African populations, those are not much affected from these allergies, but you will hear, especially from US and Europe, that allergy is a problem. Even the Indian population, if they have spend their life, basically the childhood in US and Europe, they are likely to be susceptible basically for these things. So you might have seen different papers. I will not go in detail, but what it says that if you introduce minimum level of peanuts during the childhood, different stages, so that kind of tolerance level will be developed. And there is some level to it. You can develop, means you can say 100 mg, you can develop tolerance up to 100 mg. If that is the case, and I think US and other government agencies, they are working on it, developing. And if suppose you can't do with the current generation, right? But the coming generation, you can develop, try to develop tolerance, some kind of tolerance. If you are able to develop tolerance, then also the kids should not be exposed to the peanuts with high allergen level. What I mean to say here, most of the times you see sudden introduction, sudden consumption of peanut, even little bit, it triggers. So if you have peanuts with very less allergen content, then definitely it will be better. Although it is not the solution. What we have done basically, most of the protocols we have seen earlier, those were from products, like you collect products from the market, peanut products or any food products, and then you quantify the content of those allergens. What we have done basically, we developed allergen monoclonal antibody based ELISA protocol for from the seed, groundnut seed. So basically we can see whether which variety is having what kind of allergen content. So we saw in our 300 germplasm from ICRISAT, that was global diversity collection. And if you can see, just I have put two graphs here. Left side, if you can see allergen H1, RIH1, you can see, and those values are, these are in microgram per gram. So you can see like very low level of also present. Although there is a variation between seasons, but not so much. There is some consistency. The low remains low as comparison. So if suppose if you see the right side, you see it goes to many thousands. What I mean to say, and we have also done, these are some of the varieties. I have not put that slide here. Most of the varieties currently we consume, they are having about 20,000, 10,000 of microgram per gram. What I mean to say here, we can avoid, suppose if you want to, industry has to flourish, they have market in different places in US, Europe, then they can avoid such kind of products. We have completed this last year, and then we got some of the genotypes we have done at our place. Now we are trying to get it done verification with independent labs something, and then some of the companies, they are approaching actually not as a basically for product in the market, but they want to use them in developing the tolerance to expose while they are exposing. So they want, if such peanuts are available. So that kind of discussion we are going on, but really it's a very complex problem. Okay, now you have genomic resources, markers, but again, nothing is useful for the farmers. It's all science we are doing and making information available for the public domain, but ultimately matters what we are giving to the society. Now, so this is the third phase. As I mentioned that ICRISAT, we work with advanced research institutes and the national partners. So this is the component actually, genomics enabled breeding, that we do with national partners, because they are basically our stakeholders. They take our product to the farmers and markets. How? Let's go through it. So let me mention first here markers. You must have seen that somebody has reported identified associated markers. Actually, you see hundreds of thousands of paper you see reporting associated markers. But are those being used in breeding program? No, hardly. Why? Because there is no validation mechanism, no verification. They have not validated how effective those markers are there. That is why you see you will end up very less number of diagnostic markers that are being used in breeding program. Otherwise, there are many reports on that. So I would suggest the young ones, basically the researchers who are working in this area, don't leave your research of just publishing paper reporting some associated markers and genes. Make sure you follow up your research unless your information is not being utilized by the research community, by the farmers, that goes research to the farmers. Don't leave on the others that, okay, this is not my job, others will do. I'm not sure others will take 10 times, but you can 10 years, but you can do in just 4-5 years because you know your research. So this is what we have been following, and I will show that impact as well. This is a STPG project that is funded by Bill and Melinda Gates Foundation, and that is I was talking about that in $1.5 you can get genotyping data for 10 SNPs, and that is how you can see many crops here included. Anybody can approach, anyone can use. It's open for everyone, and you can just ask those colleagues that okay, in my crop for which traits markers are available, and then accordingly you can plan and use it. How we are using in groundnut, this is the example what we have rust, late leaf spot, and oleic acid. We have put as a 10 SNP panel, and we can get data for all these 10 SNPs in just 100 rupees per sample. And that's very precise because you can get results in F2 stage, very early stage itself. We have developed this in 2018 or so, and even we have not published this paper, but you can see till now we have used 200,000 data points of this assay from Intertech. Not only us as ICRISAT, from other parts of the world they are using this one. So as I mentioned, whether we publish or don't publish, it's open and every time like basically accessible to everyone. That is the beauty basically we have at the institute. That's a really wonderful experience that you don't have any limitations to share, and it's you really become very happy if it is useful for someone. And we are trying to now improve it further by adding some more traits I was mentioning in my previous slides. What we have done basically, first we have demonstrated that how it has to be used. We have used those markers earlier, SSR based markers. These results I'm saying, what I'm showing here, it's here we use SSR markers. Now we are using SNP markers. But these are in final year of trial. You can see some of the lines performing yield also. And there is one issue I will, one important thing here. Earlier foliar disease, whenever you get such lines, they were long duration. But here we have been able to combine the short duration and foliar disease resistance together. That is the beautiful work that have been done, and then markers have helped in making selection of those regions. So that is wonderful. Now coming to the high oleic, we have used those markers, and in India we did not have actually high oleic source. So this is the genotype Sun oleic, that is from US, and we use that one, and that is a runner type. So basically we brought in Spanish bunch type that we grow in India here. What happened to those? What you have seen, two of the lines basically 15803, and you can see these are being identified and these are being released in India for cultivation. We have shared to our African partners as well, and we are hopeful that some varieties will be released there as well. Now what we have done also, we have combined this rust, LLS, and high oleic, and that is also we have done in some of the lines, high yielding lines. Those are popular varieties, so those will be going to ECRIP trials in India for release. Okay, it's not only that ICRISAT uses those markers. You can see here like the one examples we have generated in green shaded one, that at ICRISAT, but at the same time you see at least US Harvard and ICR-DGR Junagadh, they are using these markers for developing product. That means when we work, basically we work with them. Even the high oleic we breed here, that is being released from ICR-DGR. So we work with this national partner. We encourage them to use these markers in your breeding program. They should have independent program, and whatever material we develop, we share with them, and they can test and release it. Sorry. You might have seen a presentation from our Prime Minister Modi Sahab, and then he has dedicated some 17 lines, I think, to the nation on the foundation day of FAO, and two of the lines actually in 17, those are groundnut varieties, these 4 and 5. So basically this is a new era of high oleic varieties, and really I am really happy because this trait was not there in India at all, and all the international industry, they wanted high oleic products so that raw material is available in India. And really we were working with, even funding we received from Mars Wrigley, that is a chocolate basically company, you can say that as a support. So they wanted raw material actually in India, and that is how we developed, and this is now available. So we have two advantages here. The quality of Girnar 4 or 5 is equivalent to olive oil, which is around some 800 to 1000 rupees liter, and this groundnut oil is just now there is no yield reduction, so basically it will come around 100 or 100 to 20 rupees per liter. This is one advantage, that the Indian consumers will have high quality cooking oil. Now the second one, high oleic importance. If you have storage, right, all the products you basically you make, so you have to store it. So storage you can have long shelf life it has. So if it is a high oleic groundnut you are using in your products, in place of 15 days, even you can keep it for 2 months. So that is the beneficiary for for industry as well. So you see health, industry, both are happy. So that is like a win-win situation in all the aspects. And what we see from here, maybe next 10 years, I think from zero hyolic varieties, we will be seeing in next 10 years, all the varieties we grow in India will be hyolic. So this trait will be like must have trait because it's a win-win situation, nothing we are losing in that one. So that we see. This is a picture from this is from Africa, and we are seeing some of the lines also will be released in Africa as well. So these are under evaluation. Okay, the another approach is genomic selection. I will not go in detail, but because this is very complex, and just in simple way, I want to say that making prediction, it's very difficult, and it can be more precise as much good phenotyping data you have, what kind of training population you make. So you can read out some of the papers here. Only what I mean to say here, most of the traits are affected by environment, and really the models we should use those models only where the model can take care of genotype into environment interaction. Otherwise the prediction will be not of much use for the complex traits in the breeding program. We have just published this paper in August this year, and what we have done basically, we tried to see, to compare the models, 4 models basically. In the one model, main effect model, that was not having genotype into environment interaction. What we have seen basically that the last 2 models, informed interaction model and naive and informed interaction model, they have performed better. That means, suppose if you have 100 genotypes for parental lines, if you have data on 80 also, it can predict for rest of the 20. If you have 7 locations for the breeding program, if you have phenotyping data only for 6 breeding programs, it can predict for the rest of the breeding program. What I mean to say, basically, these models are really serving good. And what we are trying from next year onwards, we are going to use these models to initiate the breeding program and then basically see how these work. Until unless now, what we have seen, most of the results coming successful results from rice, from maize and wheat. So let's see how it works in groundnut. Okay, the one what we are talking about is actually one part strategy. That means you use elite lines, cross them, and then do GS models, and then you select. Here, this is a 2-part. That means it suggests that through GS, you can do continuous improvement of your founder parents as well. That means you use those elite lines. Whenever you get better lines in your breeding program, then the founder parents, you include them as a founder parent. Okay, that means you are making the founder parents dynamic. So this is 2-part strategy, and that we are trying to implement. We have one project with the Department of Biotechnology, and also we are collaborating with John Hickey, who is in the Rogeling Institute, University of Edinburgh in UK, who is basically have demonstrated how 4-part strategy is working. So we are working on 4-part as well. Although he has shifted now to company, but still we are continuing that work on those aspects. What is that 4-part? Basically, I have not put here that slide, but that means there you can use even the wild relatives. So that is the strategy. Okay, here this is the what you see initial training population. So this is I'm telling for groundnut where we are, and then testing GS models, panels, and then what we are doing actually selection. And then once we will do selection, we will go for multi-location trials. And as I mentioned, the best lines we will get here in multi-location trials, we will send them, include them in our founder parents. That means in training population. So making it dynamic. And then the good ones, of course, it will go for nomination and release. So now another concept, earlier we were using SNPs for selection. New concept is haplotype based breeding. So basically you cannot get always that that SNP is there, only this you select this one is SNP. You need to come across basically the haplotype concept. You have seen basically the very good paper from Bevan et al. In Nature 2017. And here you can see that if you combine different haplotypes, then you can get basically a better phenotype. The right side, you see that example generated in present P crop from our group, and they are trying to basically identifying the already identified those haplotypes and trying to bring together, and that is what it's a haplotype based breeding, because the genotyping may be in different way as not the SNP we do simply. So basically detection, of course, for superior haplotypes you need high quality and high depth sequencing data that we will talk later. In groundnut also we initiated, and we have sequenced 300 genotypes from globe, that is actually the representing the global diversity, and the objective is same. We have used phenotyping data on that, and we will be trying to identify those haplotypes for important traits which we have not been able to develop so far in groundnut. So although I have some preliminary results, but I am not presenting here to make it short, but this is the objective. And I think every crop now from SNP based to, there also will be haplotype based breeding that is coming up, that will be optimized in coming years. So this is the new one. You might have heard about genome editing. Yes, if you have known function genes, yes, you can do genome editing. Nowadays protocols are being optimized that you can do multiple edits in one go, but this is also now opening new areas. And yes, you can, duration is short. Again, technology point of view, I will not go in detail. I will talk applications. If we have known genes more and more, we can edit those without making any background changes. That means if a variety is very popular and you have genes for 5 traits, you can change it. Variety will remain same, and the 4 traits will be value added basically. So that is the concept, and it's less time. So that is the good thing from genome editing. Of course, it is a very less time, just 4 to 6 years you can release your material in place of 8 to 10 years. And this is very, very important. For higher genetic gain, varietal replacement should be faster, higher. We need not to grow 10 years, 20 years, 20 years old varieties. And that we can see like in India, even like we see JL24 and TMV2, this is 30, 40 years, many years old varieties, still farmers are growing. So that limits actually the productivity. Okay, there are many examples. Again, I will skip this one. Now I want to discuss this one. This is promotion of alleles by genome editing. What it means basically, right now what the researchers are doing, they are editing the genes which have larger impact or larger effect on the phenotype. Here what it does, even a smaller effect, genes is those functional genetic variants basically, if it has less effect also, if you can edit all those base pairs basically, say 100 edits, then definitely that process is called page. So what it means basically, you can integrate with genomic selection. That means you can perform genomic selection and you can edit some of these alleles and then club. So you can see this is a very good paper from genetic selection evolution if someone wants to read it in detail, because it's too technical to explain here. In summary, I will say that if you club with genomic selection, the gain, it is very high. It's a really dynamic basically. You can see you can gain genetic gain, you can really in less time you can make higher genetic gain. So that is the objective here. Rapid generation advancement, and maybe in speed breeding you might have heard. In many crops it is optimized. I am not talking about other crops. Yes, you have, if you are working in wheat, barley, chickpea, rice, canola, of course you can go from 2-3 generation to 6 generations or 4 generations. As far as groundnut is concerned, there is one, I will skip this. So basically, okay, maybe better. So basically what you can do, this varietal development process you can reduce through speed breeding, right? So here you can reduce. So basically what we want actually, the breeding process, we want to compress it so that the improved variety can be developed faster, right? We can use those approaches where you can change the multiple genomic locations and traits you can modify. So that should be the approach. In groundnut, what we are seeing, normal glasshouse conditions, you can see from University of Southern Queensland, they have been able to do 3.5 generation for a mid-duration variety in single year. At ICRISAT, we have reached to the stage, basically you can say it's the same thing, 3.5 generation in a single year. But now we are enhancing the facility further, that we can get even 4-5 generations per year. So again, this is a very good resource you can use for different applications. Suppose if you are developing a recombinant in midlines, from F2 to F4, if you can get 4 generations in a year, you can develop real population in just 2 years in place of 4 years. So that is the time gain you are. And in breeding also, if you don't want to make any selection between F2 to F6, you can go ahead for RGA, and then you can advance the generations, and then you can make selections at later stage. Although University of Queensland, they are optimizing phenotyping for some traits in the seedlings, while even RGA is going on. So if it is feasible, it's on the crop if you can do it. Again, although groundnut has advantage of being having a big seed, because seed rate is very high, because you need more seed for planting in farmer's field. So that is a challenge. If it is a smaller seed, you need just kgs of seed. That's it. But this is not the case in groundnut because seed is big. But if it is big, we have also one advantage. What we have optimized, that suppose if you have 1,000 lines to grow in the field, and we want to do marker state selection, or we want to check QC, we are not planting them in the field. What we are doing now, we are chipping them, and what you can see, the small slices here, we send for genotyping. In 15 days we get data, and then based on that data, genotyping data, we take a call that out of 1,000 lines, we will grow only 500 lines. So basically we are imposing that selection before planting itself. So also saving time and also resources, and really it's a good impact, because if you see that how selection we are doing, so we ourselves, we are how improvising. So far we have been doing, we isolate DNA and then genotype it. Also we were sending, sometimes isolating DNA and then sending DNA to some service provider for genotyping. Then we started with intertech, not DNA isolation, just leaf punching. We are sending just leaf punched those samples and then genotyping. Then come up with seed chipping. That means you need not even grow plant. You just from the seeds you can chip, you do, and once you are satisfied, then you can use. So basically you are reducing, you can see the time from 45 to just 15 days, and then dollar value, of course the money and resources you are using less. So we are making it routine, and wherever it is applicable to reduce cost and resources. So this is the now modernization of breeding programs and processes we are proceeding it. Whatever genomics we do, I will not go detail for phenotyping because I don't have expertise on that. But all our genomics studies, they are relying on the phenotyping. Genotyping, sequencing, not an issue. Still, we are struggling for phenotyping for many traits in each of these crops. We don't have a protocol, reproducible protocol, reliable protocol for phenotyping those traits. So here what I mean to say, you have 4 pillars and of genetic gain, and first 2 pillar, if you see, size of breeding program and faster breeding cycles, all those things. So basically you can see phenotyping is very, very key role. You can see it's very important, very important role. These are the like different kinds of phenotyping protocols you can assess, like from satellites to you have high throughput, mobiles, aerial, even underground also you have some of these systems. What I mean to say, as much good as the phenotyping data, as much will be the genomics studies. If you have very good robust protocol for phenotyping in your breeding program, you can avoid phenotyping and using markers. So markers will be more reliable if the phenotyping is good during the trait mapping genomics studies. So this is very clear. If you don't have good phenotyping data, you don't have good protocol, I am suggesting please don't waste your time, because the markers you identify will not work at all. Because it's an association. You put any kind of genetic markers data, you get some association. So if phenotyping data is not robust, not reliable, not consistent, then definitely you are not going, the results is not going to be work in your breeding program. That is why many of the scientists, they just leave up to there and then publication, and that's okay, I washed off my hands and then see, I have isolated, now you people use it. So that is the approach we should not have. Please make sure that whatever you do, your technology reaches. Again, decision support tools, I will not take much time, but I will say we are generating high throughput data, really big data, and those should be stored well. There is one GOBI, that is a global community, that is platform, Genomic Open Breeding Informatics Initiative. You can submit databases and it can provide linking. There are also some of the tools that they link You can do some of the analytical tools as well here. You can use, these are all free in access, and you can use it. So this is the beauty. Again, it's an integration. Your data should be integrated well. Otherwise, how the information you have, you can use in your decision making. So again, whatever you do, genomics, phenomics, and everything, it should be linked and retrievable so that you can make good decisions. Okay, if you see summary how Groundnut has come a long way, so you can see like basically 2010, if I say, and now from having just say hundreds of thousands of SSR markers, now 2-3 high quality genomes, and then deployed genomes, all the genotyping assays, everything is available. So this is like 10 years have been very good. What I mean to say here, now groundnut community is as much equipped as rice and wheat, and there is no basically now excuses for the groundnut breeders that they are away from accessing the technology as the wheat, rice, and soybean breeders are accessing it. So that I mean to say here. So we are on the same page, and then really we can make a good progress. What I would say here, genomic resources, no more issue. It's very cost effective. It's very reliable, and very less time you can get it. High throughput phenotyping data, again, it's a really, we have not been able to solve it. Really, we need good phenotyping protocols, consistent, stable, so that we can make good progress. Integration of next generation technologies, yes, of course. As soon it gets, it should be optimized and it should be used. And that's what as a CG center is our role. Basically, new technology, we just work with in advanced institutes and make sure that it is optimized and used by our national partners. International, national, and state government, private agencies, you can see most of the funding agencies, they are diverting their money for development, actually. But until unless you have research component build up, in long term, you are losing many things. So you cannot divert your attention from research to completely development, because development can be seen impact very fast, and from research it comes late. But this is a pipeline. If you break it, then this is going to be problem. So that is my message basically to the our group today here, and thank you. And then again, thanks Dr. Majumdar and Soma. Really, it's wonderful. I would love to have questions for that.
Yes, sir. We have few questions. I am just giving you in the chat box.
Okay.
This one, the first. I give you 4 and 5 questions because we need also time for the interview.
Okay. So should I stop sharing now slides?
Yes, sir. And you can start and give me 1 or 2 minutes to paste 4 questions.
Okay.
This one, the
I think there are some questions coming.
yes, sir.
In the chat, yes.
Chat box, I'm seeing actually chat box.
Yes, sir. You can start to answer, sir.
Oh, okay, okay. So I will read those questions and then
as it is
That
in
would
the
be
bracket.
great.
In the bracket, there is the name of the participants.
I think maybe
and
I will just. So here I have seen one question that says, how can we apply the translational genomics for crop improvement in developing countries? Because there is limited infrastructure. So I think maybe if you might have seen in my presentation, I clearly mentioned, for using these resources, you need not to have facility there. For example, sequencing, that's very cost effective. You can choose the best service provider. For high throughput genotyping also, you can get best price, low cost, cost effective genotyping facilities. And each crop, we have those assay developed and in public domain. For groundnut, suppose, we have developed 58K SNP assay, and it is freely available, basically accessible to everyone. They can directly contact to the service provider, they can pay and they can use it. So nothing, no restriction. And then like Intertech, that is like HTPG project. Anyone in the world can use it in just $1.5 10 SNP. So you can get access. what I mean to say here, there is no limitation. Now the technology has come that we, especially from CGR, we encourage those technologies which becomes accessible even by the breeders or researchers sitting in the remote areas, because we work with our African countries and we need this. So that our focus is more on that. Then second question, it comes in genotyping platform. What is the most advised or efficient means of trait mapping? Again, if it depends on the basically crop. Suppose if you are working in rice, wheat, you can see many chips are available. And for mapping, I will say that you can use FE or Illumina or whatever, like I'm not mentioning for advertisement, but whatever assays basically available, you can see the cost and you can use those. It is accessible to all, and you can use for trait mapping. For genomic selection, if here selection means genomic selection, then I will suggest mid density assay. That will be the best one if it is available in crops, because that will be below $10 or so, and the high density will be above $50 or $60 per sample, so costlier than that one. And if you are selection, if you are meaning by marker state selection, foreground selection, then I would prefer to go for Intertech, where you can get in $1.5 10 samples. So no need for DNA isolation. Just from field, you send samples and you get your data. Okay, now here is one question that is from Debosmita, I think. When do we use dominant marker and when do we use recessive marker, which is more efficient and why? So it's not about efficiency actually. These dominant and recessive markers, they have basically different kind of nature of identifying those alleles. It depends on the population you are screening them. If you have a recessive allele basically, and then you are doing in F2s, because I am giving example F2s because you have like homozygous, even recessive alleles, and then all those things something. So you can use those kind of, I will say SNP is the best choice irrespective of your using. These are very much working and cost effective. So I don't think that there is issue now. This was the earlier days when we were discussing about this dominant and other things, when RAPDs, RFLPs, and the others, AFLP, all those coming into picture. But nowadays, everybody is shifting to it. I don't think that is a issue. You have question, your most improved varieties groundnut that you can recommend for farmers. Again, like one solution, it does not fit for all. So basically, it depends on which region you are. So most of the varieties, they are actually being released to targeting some countries. So you can look for basically the African. Suppose if you are in Africa, we can get connected to our African breeders. They can suggest the best variety for a particular nation. If you are talking about India, again it differs from state to state. So I cannot say that I recommend one variety and then you can go it. So okay. There is one question here. Are there any genome based advanced breeding programs in groundnut at ICRISAT that can significantly reduce the number of it? Right now, I will say that there is no basically report. Basically, we have that there is some product based on the genome, but of course, this is one of the area our scientist C is working basically on that, and hopefully in future we may have some product. So because this is an emerging area that we have to work basically. But if you ask me, I don't work in this area, but this is a very beautiful area. I think if I get opportunity, definitely I would like to learn it. Again, it's a question from Tefra, I think, who asked which genotyping is appropriate, affordable, cost effective for developing country researchers. I think again it's basically again crop, and then again I mentioned three kinds of technologies. It's high density chips if you have access to. If you have access to GBS, if not, then GBS you can use it. It's not much costly. And then if you are using marker state selection, then you can use Intertech facility, because there are many labs of Intertech across globe, and you can access those labs in just $1.5. So I would suggest to for these technologies. And mid-density assay, I think in only 1 or 2 crops it is being developed. So in 2-3 years it will be developed in other crops, so will become available. So what I'm saying basically, having the best, you can just see if you crop, if you want some crop, if a specific name, you can just message me. I can connect to those people. They have something, the appropriate resources source from where you can get genotyping services. I can help you. And there is one platform that is called Excellence in Breeding. That is from Bill and Melinda Gates Foundation, and it takes care of compilation of all these resources. So you can go to these websites and access. If not, of course, you are free to contact me and I will connect to those people. There is one question that is planted in groundnut in Philippines. So of course, any plant can be planted there, but I feel there the emphasis is more on rice and other crops. But of course, yes, if you see, and farmers don't grow until they get a profit. So if they get more profit from groundnut, of course. But yes, many of the Southeast Asian basically countries, they are growing it. Myanmar, I know, basically. Yeah, I think you have to see how profitable it is for you. Of course, you can try. So now another question is like yield of groundnut. So again, it differs from region to region. If you go to US, yeah, you can get higher yield, even 10 tons or so per hectare. But if you talk about in India, average yield will come around 1.7 tons per hectare. Why? Because each state has different soil types and climate. India, although it's basically a one country. If you see states, there are a lot of diversity in that. And if you see like few states, I will name like Telangana and say Andhra Pradesh. Here you get less than 1 ton. Because of that, maybe the productivity, national productivity is less. But if you go to Gujarat, if you go to Tamil Nadu, even Rajasthan, you can get even 3 to 4 tons per hectare in groundnut. So it depends. It depends. So basically on the soil type and the, of course, the practices, field practices you do. So basically, how can we work on this in collaboration in Ethiopia? I feel in Ethiopia, I have not seen much interest for groundnut. Of course, chickpea is there. I know that ICRISAT is having good collaboration and even breeder there in Ethiopia. But of course, if you are interested, you can just contact their ICRISAT, and then definitely we can have collaboration. If you want to collaborate with me, you just send me message. So there is no issue on that. I will be happy to collaborate. Okay, I think.
Yes, sir.
No more question, I think so.
Yes, it's enough, sir.
Okay.
Thanks. There are a lot of appreciation. Your slide so simple, so colorful, so informative.
So I did not try to make it technical because audience is diverse.
Yes.
And I always keep it very simple. If somebody is interested in more technical,
because you refer the citation in every slide, there is an option to go and read more.
Yes.
So there are lot of opportunity to learn. Because as I say that groundnut, many people have many questions related to soil breeding cycle to yield, and we choose a little bit related to your talk and for today's. And I will send all the questions later after the webinar. Now I request Shoma, please do our interview, sir. It's not an interview. We are not. No, please pardon me. We are not the.
Just to know you better.
Know you. Yes. I nowadays I say it's inner view. We are wanted to view inside you, not the interview. So Soma.
Okay. All right. Let's begin the interview section of today's event. Today is our 25th webinar of the series, and we are highly honored to have Dr. Manish Pandey from ICRISAT with us today. Sir, you're quite a successful plant scientist, and we would love to know how you built your career. Could you please tell us about your journey so far as a scientist?
Uh, so, if, frankly speaking, I never thought that I will become scientist, and even when from the childhood. So, what happens? You see people around you, working people, different career options, and then you somehow start liking some profession.
Right.
And since my father was a lecturer, English lecturer, so I was really very impressed. You go to classrooms, you can shout, you can
Yeah.
just say okay, sit down, yeah, you can have lectures,
Hmm.
right? And then this is one side of it, that you are doing a job. The other side of it, that you are contributing to the society.
Right.
What I mean to say, there are jobs where you just do job and it goes to company, or it goes to is just salary, and you, it
Right.
is between you. That profession I like because it has a different angle. You are getting salary as well, and then also you are contributing to the society, and of course teachers get lot of respect as well. So basically I was seeing from childhood how it
Mhm.
is, so I start liking that one. So when I grow up, then I see like in fifth standard or eighth, I saw teachers, oh, how they were like basically managing students. Okay, whenever they want, okay, you sit down, you do this. When I became came to intermediates or graduation, then I thought these teachers are much better than those in basic schools. When I came to post graduation, so of course, then you start
Mhm.
and you talk about science and then research. Then I come to know, okay, there are also researchers,
All right.
and I have also an option for research, but I don't know how it is going to be. I have never met those scientists or something, whether
Mhm.
I'm going to take wrong decision. So just I took a very safe side, that okay, let's
Mhm.
do PhD,
Mhm.
do some good research, and then think whether you want to go to teaching or you want to go to research. So you
Okay.
can say basically from there onwards, like I will mention names of say three mentors, like post graduation I was doing in Allahabad. So basically Dr. Chitranjan Kole, where
Okay.
basically he just guided how genetics is the core of biology, and then how it impacts the entire functioning of plants and animals,
Mhm.
and how lot of improvement or scope in career. Then I came to DRR like Hyderabad, and during PhD Dr. Enso Barani. And then after post PhD, when I landed up in ICRISAT with Dr. Rajiv Vasne, you might be knowing him.
Yes,
Uh,
definitely.
and really he's like basically I have different personality, and then I come to this world researcher. So even after PhD also, I came here, okay, I will go for postdoc and then all those things. But when I have seen the environment here, I thought that okay, I will be doing some job in US, or I got opportunity as assistant professor in Raipur in one
Mhm.
university. But I choose an ICRISAT as a role because here I seen two advantages. One, there is no limitation, no uh, basically no one is to stop you to gain information and knowledge. You can collaborate with everyone. There is no limitation in dissemination of your knowledge and products and technology. So basically, in addition to India, you can contribute to several other countries. So that is how
Mhm.
basically I landed up being a researcher in ICRISAT, and then being in research. So I love more in research now. I have moved basically teaching from mind time being now. So I'm happy with that. And many of the colleagues, if I met my bachelors and others, they really surprised and say, I can't believe that you will be going to research because you have never been mentioning about this career.
Okay. It's wonderful to hear your life story. Sir, you have, how have you entered into groundnut research? Could you please highlight a few key successes you and your team have achieved so far?
So actually did PhD in rice, and then after PhD, we see that okay, now let's have some postdocs and have different experiences. When I applied, I was looking three options. One was in ERI, and then one was in ICRISAT, and one is private company. And when I came ICRISAT, actually I never seen groundnut crop before that. So even interview, Rajiv Vashne asked whether you have seen groundnut or not. Then I said no. Then backside it was a picture of groundnut. He said, okay, I will show you right now. Groundnut in picture at least. So that was basically the one. And then I entered in groundnut. And the good thing was from where I left in rice, there was a need to start from there in groundnut. So basically that is how my connection.
All right. Sir, your team decoded the complete set of chromosomes of two widely cultivated peanut varieties. How does this genomic information benefit crop development and the field of research?
So basically, if you see that we have actually two species I mentioned, botanical groups. Actually, those are like Spanish bunch that we grow in Asia and Africa. You see little bit straighter type. And the runner one that you see basically in US and Europe, in Australia as well, runner. So we have genomes of both kinds of both species. So when we are doing analysis, especially if we are sitting at ICRISAT, we use these relevant genomes for doing all our genomics analysis. So like these, I will say, okay, in simple example, for breeder, they need founder parents to make crosses. Then only they can develop some variety. For a genomic scientist, actually it is like a basic founder, basic material, basic resource, mother of all genomic resources, from where all the research starts.
Yes.
You take markers, you take genes, even if you want to do gene editing precise location, if you want to confirm, so everything is related to that. So how we are using it basically, we are using now sequencing based trait mapping. Like I mentioned QTLC, WGRS. So this is how it is helping us in those. This is one, gene discovery and marker development, and genotyping assays. Suppose we developed a high density genotyping assays, 58,000 SNPs we took from that genome. Then again, we are developing mid density. Again, we are taking best 5,000 from the genome by comparing sequencing of different genotypes. So that is how it is helping. So basically what you see work molecular breeding product that is going to the farmers, the markers used, these are coming from these genomes. So size location, that's very important. So.
All right. Sir, you have received many fellowships and awards and do quite large collaborations. Sir, what is the driving force for your dynamic nature?
I think I never thought of this basically, although I have met with very high profile researchers, but because I was started with PhD. So one thing I was having in the mind that you do little, but do the high impact. Actually, if you do lot of things and then it is repetitive and maybe making less impact. So what happens? Science, you get some recognitions, but you are not feeling happy from inside because you are not contributing to the society. For example, like when I came here, basically like genome sequencing, all those, these were required for research community. And then there was no high oleic. So basically when a company came to us, they said we want high oleic, but we don't want to purchase or import from outside because it will become so much costly. We cannot compete with other companies. So we said, okay, fund us, we will develop material, but our condition is there is no patent, nothing like that, nothing, no protection, nothing, no limitation, anybody can use it. So this is always our condition. We work with fund from private company, this is our condition, that the material should be freely available to everyone. So I feel once you do research, then you feel that many people will like to connect with you.
So true.
So I will say initially this was that we were connecting with people, okay, together we can make this impact. But later on, we realized that many people also they are coming up to connect with you. That's really gives happiness. So And once you have some research and all these achievements, of course, these fellowships and awards will follow. So I see as a byproduct of or basically recognition in terms of these. But again, the crux is that if you focus on research that can make high impact and make sure that it reaches to the customer whom you are doing research. So that's the basically what we are following. And that is why we take risk also sometime. And when I started here, like say Largen, oh my God, was a huge discussion that it is not a solution. I said it's not a solution. I agree. But a person, if it is that is exposed with 25,000 microgram per gram, if we can give a genotype just 10 microgram, I don't know how much impactful it is. I don't know how it is used, but it will be good to have.
Right.
So that is how we pursued, and I'm happy that some companies at least they were showing interest and then saying, okay, it's a good stuff. We were not aware of this, that there is a diversity, so much diversity for allergens as well. So that is how basically you come up basically, and really in science, if you are ready to fail, you will never fail.
That's a good one. Sir, you're also an adjunct associate professor at the University of Southern Queensland, Australia. Sir, is the method of teaching or instruction there different from that of India? And how is your teaching experience there?
So basically I don't have any teaching assignment. So how I got connected with USQ is because of work. They had basically they wanted somebody to guide or basically support the genomics research for their students. That team. So I got like basically letter that we want you to be a adjunct faculty here, and we want your support in completing or doing PhD research and then proposals, collaboration that we do. So because of that, I got, and then I got associated. I have two students, joint students from there, and they are doing on different aspects. So I'm supporting my expertise from here. Likewise. So basically, I will say it's not teaching.
Okay.
It's completely on the research, and that is on the, because I have to dedicate more all the time for the ICRISAT.
Right.
So that is why I have restricted basically to the research, which I am comfortable doing so.
Okay. Sir, according to you, which sectors of plant science research will prosper in the near future?
Actually, what I see basically in last 5 years or so, every researcher is generating huge data. It's phenotyping, it's genotyping, it's sequencing. And I'm seeing that for this data science is really going to be a crucial phase.
Oh, yes.
And really, I see a great career opportunity for young ones, I will say. I will give you example, I will not name it. I have seen that even few researchers from different countries, we can't do coding, but even they're from normal biology background, they can do coding. They can write scripts, small scripts. What I mean to say basically, from we have to basically reboot our basically those postgraduate and PhD time, and then analysis data. That area is really, without that, I think nowadays our research is really incomplete until unless you are doing total even lab oriented, even wet lab work also. You need a minimum statics, we need minimum analysis to put up your research. So that is, and that is how I am encouraging my student as well. I say very simple thing, saying to the saying that if you can't eat the sweet of your own soap, that means you are not making good sweets. So I say that until unless I believe that I am eager to continue after PhD. Then till then I'm not satisfied that I have done justice or you have done a good job being a student, because you need to have some edge nowadays in career and all those things. So I just encourage them. Even first year and second year I put some kind of encouragement and pressure. Okay, you should learn this, all those things. But again, it's up to the student. I say you can't make them like, they should become efficient. But some of the students really, which are self-motivated, I see, they always pick up. So that is a good thing.
Sir, many of your publications showed a strong international collaboration. So, what do you do, or how do you successfully maintain it?
So the best mantra is we don't compete.
Okay.
I will give an example. Suppose if you go some of the meetings and we have ideas, we openly share it. And if somebody comes and we say that we want to do it, and whoever wants to come with us, we are happy to work with them. Many times people come, hey, we want to do it, but we want to lead it. Say them, okay, you lead it. Because our job as a CG center is the core activity that things should happen.
Right.
We don't want to take credit that, okay, we want to lead this, we want to lead this. We want that this should happen, and the science and the agriculture that we are into, it should progress. That is our main job. So we never, even with national partners, even with proposals on I write down just objectives and discuss with them, and just first I say, you pick up the objective you want to do at your center.
Right.
Where you want to lead, you just put co-lead, and many times they choose, and then we put a proposal like that. So, and in research, we have formal, like in projects, most of the research hour is informal. Not in the project, just we discuss sometime, okay, can we do together this, can you support this, all those things. That is how it is happening. That is, and because it is a like CG organization, so we don't have any limitation to collaborate with any of the country because of the political regions also.
Right.
So that makes, and the other countries also, they don't hesitate in contacting us. So that is basically that is the good thing. And the most surprising thing is, many of the times we are collaborating sometimes that one collaborator this side, another this side, and we have to make really balance, then nothing is repetitive, not telling things also, or basically discussing and then disrupting things, but making sure that we play their like cohesive role. If it is like overlapping, we try to bring them together, not to do separately. And if it is altogether different, then there is no issue. So many times, yes, there is a problem, but they understand us well. So that is why many times they approach us, because these people will be having information whether this such kind of work is going or not, or something, whether I should lead or something do or something like that. But they have been really, I should give really credit to our partners. They have been very open to us, and we have been also very open to them in collaboration in any form. That's how it's moving.
All the best for the future also. Sir, according to the Stanford University 2019 report, you are one of the top 2% highly cited researchers in the world. Could you please give some advice on the factors that we should keep in mind while approaching or submitting manuscripts in a journal?
So yes, that is surprising to me. Basically, it was in 2019 year.
And many congratulations for that.
Thank you. So I thought it's just 10 years for me, and I was not expecting actually any kind of such thing listing the top 2% also. Regarding journals, actually we choose based on the journal and the audience and our work. Frankly speaking, first stage we don't give any value to impact factor.
Okay.
We see this is our work, it fits to this journal, and in this category we have 4 options. Then of course we see the quality of our work, and accordingly we approach to particular journal. And most of the times, or all the time, we pay those publication charges as well, so that it becomes open access.
Right.
When it is open access, you might have seen that then people will cite more, read more, and then use information. So I feel that is one thing, that if we can publish in related journals, you know who the readers are, then only they will read and use it. And then open access, I think you should, if it can take care, I think to take care of those publication charges. Else, you should publish in those journals where they support open access or something. I think so. And I don't believe basically in impact factor. I say if your work is of high impact, no one can stop readership and citation.
That is right, sir. Sir, it was wonderful talking to you. And as a conclusion, could you please say some words of advice to the young research fellows who are watching you and getting inspired by you?
I think that's a very tricky question, and many may be unhappy with me by listening. But what I mean to
No,
say,
no, no. They're not unhappy, sir. They are appreciating, like everybody is showing appreciations on the chat.
so, Soma, what is happening basically? Teaching is all together. Many young researchers they mixed up with teaching and research, and
Right.
teaching is altogether a different ball game where if you secure your job, you just keep on teaching and then job is done. Research, it's a 24 hours job. You have to keep updated proposals, research. You have to be on your toes, basically in forefront in the areas you work. So I feel if you are ready, a young person is ready to take that challenge, then only they should come. Otherwise, what is happening after few years, they, start like basically demoralized or something like that I'm losing the race. So they should not feel race. That is why I said research, some jobs are there. If you enjoy, you like, you love, then only come make it career. Otherwise, no. Otherwise, you can have teaching. Of course, that is a good career option, but research means you should be ready for 24 hours on the tools. And then, of course, it is a really enjoyable. Once you start enjoying, then there is no stopping.
Yes, definitely, sir. It was wonderful watching and learning from your presentation, and many all of our viewers, from around the world, they've appreciated, and all of the appreciations are in the YouTube chat. Sir, we will send you every other details of that. Shubho will do that, and let's go back to Shubho now.
Thanks, Shoma. Thank you, sir. When you I always love this interview session. It's something. It cannot be visible until you write your autobiography. So so it's our way to collect few informations, because I found it's always motivational. It's always motivational from your talk, and I also inspire few of things like. Say when we start the bioengine, now few of others try to build the bioengine. Then sometime our team tell that they are also start the bioengine, and I always say we are both serving the society, and we are both going to the promotion of plant science. If they are working in another way, it indirectly benefit my aim and my perspectives to the science. And that's the reason, as you say, they don't need to competitions, because we are free for all and open to all. So we are not gaining it. So there is no reason for competitions at all. And sir, one thing is when we are in masters and PhD, we always feel this gap that if it is possible to reach. I pasted two another question for you in the chat box.
Okay.
It shows the relationship of this type of webinar and our platform.
Where I can see, so I could not see your questions.
No, sir. There is in a chat box. There sir. Currently, I am working on the PhD on common beans.
Okay.
Yes, last two.
Okay. I think common bean. So basically, yes, we have collaborators because it's a legume crop, and then we have a strong network on legumes. So of course, I would suggest that you contact me through mails, and then I will connect to those people and ensure that your work is done, and that's a good. And That is why I see, like, in research like as I have seen this question. See, that means I think the message is from a PhD student, right? See, this is the way that if you have some problem, the question has come how to get the solution. That is the science about.
Yes.
That is how we should not discuss problem much and all those, keep it and go for solution. This such kind of approach by researcher, that is really highly appreciated. Yes, please contact me in my mail. I will connect you those people and ensure that you get the right platform for genotyping and also analysis. If you required something like that, we will support those as well.
Yes.
Another is how I can work with you. Of course, I would suggest I work in Groundnut. So basically, you have limitation that whatever work you will be doing, that would be in Groundnut Genomics. How you work, basically, that means either you should have a fellowship. I'm not sure from which place you are. If you have some fellowship, they can work, or if we have some vacancies in our project, time to time we are advertising. So these are the two methods. If you are already a researcher or a permanent regular faculty somewhere, if you have some program for collaborative or coming here, so we can support from the projects or like that, some opportunities. But I will say you just contact me and then I will see the possibilities, how you can work with us. So
Yes. I like this type of question too, because it's remind my situation in back in MSc and PhD, that someone doing so many things in Groundnut, and I'm doing these things. That's why I always pick this question. I'm not never saying to the speaker, but I like this type of question too, because it's real. It's actual reality. When we connect that our lab can do that, this person can do that, and they can achieve nature science and nature biotech. So it's the possible, and it also shows because we plant scientists are very limited. People are very few are the plant scientists, and it is a round world. So somehow they can connect with you in near future, or maybe we don't know. We can cross paths each other in many times in that life. So it's a platform to give us, to interact us, and motivate us. What I wanted to say, sir. Sir,
really, I think you are doing a fantastic job. I'm saying basically, I have come across first time basically such kind of like organizing this, like continuously you are having from different expertise, and then actually you are connecting to the people, those who need, and then basically they don't have either resources or maybe they need little guidance. Because we also remember from our PhD days also, we were not resource rich also, something like that. And always we thought that, okay, we should have done this one, but we were unable to access those. But today's world is actually changing. We can approach, we can access those technologies and make our science basically of high quality, and that is good opportunity. Yes. Of course, through your platform, I think really, that is why I mentioned, right, as a researcher you do job, you get salary, and basically you are doing for society. You are through this platform, I'm not so you are not getting any salary, still you are doing for the science.
yes.
Right. So basically it's a, I think that's a really dedication, or really is for scientific society.
Yes,
Right.
sir. I think it is a one type of initiative that in long term it can build a good platform for India and Asia. So that's why I think I should start early. If I wait for more years, then why I should lose these years? So that's why, and it not give me any monetary benefits, but it gives me a lot of connections. I need these connections much more than the money. so so I think it's a it's a relationship to all the viewers, all the speakers, and it's connected each another in a way that sometimes
it brings us together.
Yes, brings us
Yeah,
together.
sure.
And
Because 1 or 2 times, actually, I will not name it. Some of the companies, they ask that, okay, you have presentation or something like that, and then some of the technology use that side. So basically, I differ from that. Basically, I will show my science, what I am doing, whether it involves your technology or not. That is how basically
yes,
science is. So
yes,
that is why I loved this platform. That
Mm.
irrespective of everything, you just connect with people, present whatever work you have, and then it's like a candid chat here. Like basically, how we are in the field. So each of us has some different story that has brought us to this stage, and that's the journey.
yes, sir.
Right.
Sir, now I wanted to conclude by a comment from the YouTube. Nowadays, I'm collecting the best comment from the YouTube. I'm not asking to anybody that this was the best, but I'm collecting few comments. One comments from the Suman Jha. Great, Dr. Manish sir. You are honest to the research, which delivers benefit to the society. Thank for your nice, simple, and effective presentation. So it's from our end also, sir. It was a great and very nice presentation. It was nicely collaborate. You not only give us the overview of your research, you also represent ICRISAT research and their aim. And I'm sure it will benefit more people. More people will interested in this type of crops, and there are a lot of things happening in nowadays, and in future there are a lot of scope for genomics, lot of scope for breeding, and many more. So keep working. I congratulate you your success, and I want and I wishes you that you can achieve more and more. Thanks, sir, for joining today. It's our pleasure to have you in this platform, and definitely all the viewers will appreciate your presentation.
Thank you to both of you for bringing me here and introducing to this new platform.
Yes.
It was a nice experience, and thanks a lot.
Thanks, sir.
And good luck to all of you.
Thank you
Thanks,
so
sir.
much,
What
sir.
you are doing good job, and I will suggest to keep on doing, serving the scientific society in different manner. And this is one of the strong platform and ways for dissemination and connecting people. So really appreciate it.
Thanks, sir.
Thank you, sir.
Thanks, sir. Namaste. Thanks, Shoma, for connecting with us. Sam, thanks, Pallavi, for helping to collect a few
Happy to
questions from
help.
the YouTube. Thanks, everyone, for joining today. This was our 25th webinar. I already shared our password, that is 252525, and I already got about 500 application for the certificates. And this link will open for more 2-3 hours. So don't worry, take your time and apply for the certificate if you want a certificate. Our next webinar registration is ongoing. It's in 19, and we hope we bring more such good speaker, renowned speaker in this platform in the future. So thanks. Bye bye, everyone. Sir, you can sign out. Pallavi, you also can sign out. Soma, stay 2 minutes. I need to one thing to record again.
Okay.
Before that, I stop the streaming.
Also, you can
Yes,
about the discussion session.
I will send the email to everyone.
Okay.
Not today. I stop the live streaming now, but